Today we'll be focusing on Target 15.5, take urgent and significant action to reduce the degradation of natural habitats, halt the loss of biodiversity, and by 2020, protect and prevent the extinction of threatened species. The indicator that measures progress towards this goal is the IUCN's Red List Index. The Red List Index encapsulates the data of the IUCN's Red List into a graph, with each red line representing a taxonomic group's average risk of extinction in the near future. The blue line in the middle is the average risk for all recorded species. The graph rates each group on a scale of 1 to 0, with 1 being least concern and 0 being extinct. Currently, all the lines are going downhill, signaling a major decline in global biodiversity. One key factor contributing to this decline is mining. It is unrealistic that we'll be able to put a complete stop to mining in the near future. Instead, we must focus on mitigating mining's negative impact on our environment. But what are those impacts? Chelsea will tell you more.
Mining is a significant contributor to global biodiversity loss, polluting waterways and causing grave damage to our ecosystems. Acid mine drainage due to mining greatly alters the pH, which therefore makes waterways inhabitable for many aquatic species. Waterways above a pH of 9 have the ability to denature cellular membranes. Young fish are extremely susceptible to a pH below 5, therefore forcing fish to move, altering the food chain and lowering the biodiversity present within that certain area. The neotropical region is under threat from both legal and illegal mining. The area houses the most diversified continental ichthyofauna, representing more than 75% of global functional diversity. This region is home to many endemic species, with almost 10% of freshwater fish species considered critically endangered, or vulnerable, according to the Red List Index, and therefore must be looked at. Crude oil spills are frequent within the area, with over 400 recorded in the Peruvian Amazon over a 19-year period. On July 16, 2000, a pipeline accident occurred, spilling 4,000 cubic meters, causing great fish mortality as it eventually reached the Gaki River Basin, where over 50% of species are endemic. Effects of exposure to crude oil includes hindering fish swimming and causing malformations, disrupting ecosystems and impacting fish diversity. Gold mines within this region often use cyanide, which in waterways can cause a multitude of negative effects leading to biodiversity loss. This is observed in Mexico and Honduras, where cyanide within waterways killed young fish, severely impacting fish populations present. Cyanide can also impact plants and macroinvertebrates, impacting the food chain. Erosion and pollutant contaminated siltation in nearby waterways can occur due to mining. A study in Suriname revealed that mining-induced erosion affected the water quality, altering habitats for fish feeding, reproduction, and development, along with gill function, overall negatively impacting fish diversity.
It is important for the surveys to be accurate because the information they gather are used to complete the Red List Index. eDNA is a biotechnology that allows for more accurate detection of the species present in an environment. As an organism interacts with its environment, it will unintentionally shed some of its cells. eDNA is the process of collecting DNA from these shed cells found within the environment. The following is a simplified process for studying the DNA of multiple organisms from the same environment. Step 1. Collect. A sample of substrate is collected from the environment that is being studied. This is as simple as scooping some water or dirt into a jar. Step 2. Separate. The cells need to be separated from any unwanted material also picked up in the sample. This is commonly done by using filtration and centrifugation. Step 3. Extract. The DNA needs to be extracted from the cells. Although DNA from the nucleus can be used, researchers more commonly use mitochondrial DNA. A series of chemical reactions are used to break down the membranes of the cell and organelles and separate the DNA from structural proteins holding it together. Step 4. Amplify. By using a process called PCR, polymerase chain reaction, the amount of DNA molecules is increased or amplified. The small sequence of DNA called a primer is added to the sample. If the primer comes into contact with a corresponding DNA sequence, it will bind to the DNA and trigger a chemical reaction that copies the DNA. Step 5. Sequencing and barcoding. The DNA is threaded through a high-tech computer, one strand at a time. This computer will record the order of the nitrogenous base pairs in each DNA molecule in a process called DNA sequencing. Researchers will often focus on one specific gene that can be used as a DNA barcode. This barcode is compared to a library of DNA sequences for different species. If a matching barcode is found, the researcher has identified what species of organism the DNA belonged to.
Biodiversity loss is heavily driven by mining activities, requiring immediate intervention. This is evident in the neotropics, with the IUCN's Red List Index highlighting the widespread biodiversity reduction. While mining's contributions to technological advancements is undeniable, it is important to establish a balance between technological progress and preservation. To meet the UNSDG's goal 15.5, we need stricter mining regulations, eDNA monitoring, and a global pledge to protect ecosystems.